just have multiple elements, okay? So now it turns out that these individual elements can have multiple binding sites, right? So it's not one element, one binding site, one regulatory protein, but you can have multiple inputs, multiple transcription factors binding to these elements to add to this combinatorial effect, okay? All right, then the converse of that is where you have one protein regulating a battery of genes, right? So before we were looking at one gene regulated by a battery of proteins. Now it's the converse of that. So in this case, something responsive to a steroid hormone is going to turn on all these genes necessary for, God, I can't read it, starvation, and then, what does it say below? Enzymes.
Physical activity.
Thank you. Yeah, okay. So these are just glucocorticoid response elements. All right? So along those lines then, you can have then a master control gene, so-called. And this was coined, in the case of this fly eye, by Walter Gehring in the lab next door in Switzerland. He liked to talk about the master control gene. And so MyoD is in that category too, where fibroblasts in these myoblasts have similar, they have this whole repertoire of transcription factors expressed, so that you just need to add one, and then you can trigger the fibroblast into the muscle cell differentiation pathway. So, so that's like a really key concept. So the fibroblasts are just missing MyoD. Throw it in, and you can convert them then into this different lineage. And the same thing then with the fly eye. So you can, in this case, it's not a single cell type, but an organ. So you can produce a whole organ by throwing in this master control gene, which is, it's actually PAX6. So you learned about PAX9. It's eyeless, okay? So point being master control gene. Board questions, these are pretty straightforward, right? So this, you know, we understand this. So this is the Griffith experiment, right? So the rough, and that's, he transformed, right? And then what are proteins rich in basic amino acids? I guess we didn't really talk about that too much. Histones. And then what's the function? Make compact DNA. Okay? All right. Is this good? Does this work? So, you know, it just saves you from going too far. Just